In this continuation, we are going to look at epidemiology and transmission. They are linked concepts. Epidemiology is a branch of science that deals with the spread of disease, specifically what factors affect how a disease is distributed and spread throughout a population. Epidemiologists can look at all kinds of different diseases, such as cancer and autoimmune diseases, but since this is microbiology, we are interested in infectious diseases. And infectious diseases can be transmitted from person to person or from animal to person or somehow transmitted between people. So therefore, epidemiologists are also going to be interested in how a particular disease is transmitted through the population. So that's what I mean when I say these concepts are linked. So let's quickly define epidemiology. And once again, we are going to be looking at infectious diseases. So when we look at infectious diseases, we're interested in specific numbers. We want to be able to quantify how disease spreads. So two numbers that epidemiologists are absolutely interested in are called the incidence rate and the prevalence rate. The incidence rate is the number of new cases reported in some given time frame. And usually this is within a specific area. For instance, the County of San Diego maintains their own incidence rates, meaning that they take information submitted by clinics and hospitals and other testing sites, and they compile them and can create basically a number of new cases per day, per week, per month, per year. The time is somewhat arbitrary. It's determined by the epidemiologist. If it's a disease that is spreading rapidly, like COVID-19, you might be interested in the number of new cases per day. But if it's a disease that's pretty rare or slow spreading, then maybe you're interested in the number of new cases per month. Now, one thing about the incidence rate is that the numbers are always reported per 100,000 people. And this is the way to normalize it across populations of different sizes. And we'll take a look at a real example of how we can change case numbers into an incidence rate reported per 100,000 people in the next slide. The prevalence rate is a similar type of measure that looks at a snapshot of the population at any given time. And it's basically saying how many people are infected with that disease right now. In other words, how many active cases are there at this very moment in time. And likewise, it is always reported per 100,000 people. Let's take a look at some real data to see the difference between the incidence rate and the prevalence rate. So here I actually jotted down real numbers that I got from New York Times and this CDC reporting the San Diego County case rates on particular days. So I picked numbers when our case counts were really high. So this is shortly after the new year. And I picked a week of data. So I started with January 2nd. January 1st was a holiday and nobody reported case numbers on January 1st. So January 1st positive tests and January 2nd positive tests got grouped together with January 2nd, which gives us this anomaly. So that's really 2 days combined into one. And then you can see the rest of the days. Now, if I were to present this, I could do a daily presentation. I could say I'm interested in what the incidence rate is per day, or maybe I could do per week. So if I add all of these numbers up, and this represents a week's worth of new cases, I would get 24,917 new cases per week. So one of the things that I would need to do to formally present that number is convert that number into number of cases per 100,000 people. So here's basically what I would do. I'm going to take that number, I'm going to say we have 24,917 new cases, and that is per week, and that is over the population. Now, the population of San Diego County is estimated to be 3.34 million. So what I need to do is I need to set up an equivalency like so. In other words, I need to somehow change that ratio into X per 100,000 people. And again, the per 100,000 people is a way to normalize across geographic regions that have different population sizes. So if I solve for X here, what I actually get is 746 cases per 100,000 people. So that represents the incident rate for the first week of January in San Diego County. Now let's take a look at what it currently is. So the most recent data that was presented was yesterday, and there were 422 new cases of COVID-19 documented in San Diego County. And so once again, if I want to ask about the incident rate, and this would be per day, not per week, then I would have to take that number of cases and divide it by the number of people in San Diego County, and then normalize that to 100,000. And if I do that, X is 12.6. So 12.6 new cases per 100,000 people per day in San Diego County right now. Now, everything that I've done so far is the incident rate, number of new cases. Number of new cases is relatively easy to detect as long as there is routine testing of that disease, and right now there is routine testing of COVID. Prevalence is a little bit more challenging for some diseases because prevalence says right now how many people in San Diego County have COVID at any stage. And that's a harder question to answer reliably because there are people at different stages of that disease. Of course, there are people that are infected that don't know they are infected. Maybe they're still in the asymptomatic stage. We know that to be the incubation period. Maybe they're just starting to get some symptoms, but they don't really suspect it's COVID, so they haven't gotten tested yet. That would be the prodromal period. Maybe they've already gotten tested like 5 days ago and they're recovering and they're in the convalescent period. Those would still all be considered active cases. And so that's a harder thing to get a good number on. But the estimate of current active cases in San Diego County right now is about 4,522. So if we use that number, we can estimate the prevalence. And when we do that math, X is 135. So the prevalence rate says that if we go into San Diego County today, right now, and we randomly grab 100,000 people, we expect there are approximately 135 active cases of COVID within that 100,000 person population. So now that we've finished looking at some numerical ways to quantify the spread of disease, let's take a look at some terms that are applied to how disease is spread. So when we think about diseases and the way that they are spread, there are some names that we can apply to these diseases. And again, I'm specifically referring to infectious diseases. One term that we might use is endemic. An endemic disease is going to be one that is geographically restricted to a specific area. And there is no better example of an endemic infectious disease than malaria. When you think about malaria, you don't think about the United States. You think about tropical areas, areas that have mosquitoes that transmit malaria. So endemic diseases are geographically restricted. Some diseases are geographically restricted and some are not. Typically, endemic diseases are ones that fit one of the following criteria. Either the true reservoir of that disease is endemically located, meaning it's geographically restricted. And by true reservoir, I probably mean another animal, for instance. So Ebola is not something we're probably going to get in the United States endemically because the reservoir for Ebola is not in the United States. It is thought that the reservoir for Ebola is probably a bat, and specific bat species that are found in areas of Africa. Or that particular disease uses some sort of biting insect or some sort of other animal to transmit that disease, like a mosquito. And mosquitoes and other animals also tend to exhibit specific geographic restrictions. So we have mosquitoes here, but the mosquitoes that we have in San Diego are not the same species that are able to transmit malaria, and therefore we don't have malaria here. Another term that we can apply to disease spread is epidemic, and this is one that most people have heard of before. In fact, COVID started as an epidemic. And an epidemic is going to be a rapid increase in incidence rate. So whenever you have a rapid increase in the incident rate beyond what is typical for background or basal level, that is called an epidemic. So epidemics happen all the time. We have seasonal flu epidemics. They tend to be located to a region. Now the region can be small, like a single hospital or a single town, or the region can be large, like maybe half of the United States. A pandemic is going to be an epidemic on a global scale. So here, before we get to a pandemic, here you can see a graph showing the 2014 West Africa Ebola epidemic. And what's particularly interesting is when you look at the new cases per week, you can see the rapid increase. So the rapid increase is required to call something an epidemic. Now, whether this term was in people's lexicon before this pandemic or not, I'm not sure. But I think everybody at this point has heard the term pandemic and associates it with COVID-19. And simply, a pandemic is a global epidemic. We are currently experiencing a pretty devastating pandemic, devastating both in terms of lives lost, but also in terms of disruptions and how we live our lives. The last pandemic that we had was in 2009, and it was the H1N1 influenza variety. That was also a pandemic. Before that, the last major pandemic was HIV, and that was spread in the 1980s globally. So pandemics don't happen as nearly as often as epidemics, and when they do, they can be quite devastating. So let's go ahead and define these. All right, if you need to, pause the video to record these definitions. And now we're going to take a look at transmission. So transmission. Transmission is about how does infectious disease spread. typically from one host to another. However, it's not always that straightforward. Sometimes there's intermediates involved as well. So in order to answer this question, we first have to ask a different question. Well, where are the pathogens? In other words, if I want to understand how something is transmitted, I have to first understand where that particular pathogen is or tends to live. And we call those reservoirs. And there are three major types of reservoirs. The first one, and the most common one, is simply us, humans. We are reservoirs. We are reservoirs for all kinds of different infectious diseases, especially human-adapted diseases. Strep throat, pneumonia, meningitis, Clostridium difficile or C. diff, basically almost everything that causes diarrhea, staph infections, malaria, HIV, now COVID-19. COVID-19 is definitely human adapted. It wasn't in 2019, but it is now. So humans are usually the default reservoir. When you get sick, you usually get it from another human. But there are other places as well. And one other reservoir or reservoir group is other animals. So there are lots of infectious pathogens that actually live in other animals and will occasionally, or sometimes often, jump into humans. So examples of diseases that use animals as reservoirs would be the original coronavirus before it jumped into humans. And so far, this coronavirus has jumped into humans at least 3 times that we're aware of, causing the outbreak of SARS, the original one in 2003, MERS, which was Middle East Respiratory Syndrome. I believe that might have been 2005, but I'm not sure. And now, of course, the most current one, COVID-19 or SARS-2. And this one took off, became a pandemic, and is now human adapted. So the virus that is circulating from human to human causing COVID is definitely a human virus and is no longer using an animal as a reservoir. But initially it came from an animal. Lots of other types of flu or influenza use animals. Plague uses an animal. Ebola uses an animal. West Nile virus uses an animal. So these are all examples of infectious diseases that will jump into a human, but typically is found first in an animal and uses that animal species as its major reservoir. And then the last type of reservoir that we can encounter is one that I'm just going to call non-living. This would be like pond water, soil. So examples of this one, cholera. Cholera is an ocean bacteria, actually. Vibrio cholera is found in the ocean. So water is the non-living reservoir for cholera. Legionella pneumophila. So I used that example before, Legionnaire's disease. That one also uses a water supply. So again, there are some pathogens that are going to be using non-living reservoirs. There's not a whole lot of them. Like I said, the most common is humans. Humans are the most common reservoir. So now that we understand what the reservoirs are, again, this is going to be the major or dominant home for that particular pathogenic species. Let's take a look at transmission. And transmission is basically how does a pathogen, be it a bacterium or a virus, get out of one host and into another. And in order to understand that, we need to look at something called portals of entry and portals of exit. If you are a host, a human host for a particular pathogen, it gets into your body somehow, and we call that the portal of entry. Now let's think about the disease that has been on everybody's mind, and that's COVID-19. If I get a little bit of COVID-19 virus and I rub it on my skin, that's not going to cause disease because that's not a portal of entry for COVID-19. Rather, a portal of entry is going to be my nose and my mouth. So in order for me to actually get COVID, I have to get that virus into my nose or into my mouth, and those are going to be my portals of entry. If it goes anywhere else in my body, my eyeball, my skin, in between my toes, under my nail beds, in a cut, it's probably not going to give me COVID. Likewise, portal of exit is how a particular pathogen exits a host in attempt to get to a new host. So this is probably best done by example. So you can see I have a bunch of examples here, and we're going to quickly go over portals of entry and portals of exit so that you can see the trends. So I'm going to start with strep throat. Strep throat, of course, is, if anyone's ever had it before, makes the back of your mouth feel like you're dripping battery acid and somebody's cutting it with little razors. Very painful, caused by bacteria. So portal of entry, how did you get it? Well, you get it by going into your mouth. So the portal of entry is the mouth. The portal of exit is going to be saliva, which is also coming from the mouth. So portal of entry is the mouth, portal of exit is going to be saliva. So now let's take a look at cholera. For cholera, this is going to be a digestive bacterium that causes very watery diarrhea, and it can dehydrate you to death if not treated. So portal of entry, as you can see in this image, is going to be the mouth. The mouth is the portal of entry for a lot of stuff. Portal of exit. Well, cholera is now going to go through that person, and portal of exit is going to be feces. So fecal matter would be the portal of exit. That's the body fluid that cholera would be exiting in and trying to find another host. Coronavirus, SARS-2, portal of entry is going to be either the nose or the mouth. And I actually think the nose is probably a more common portal of entry than the mouth is, interestingly enough. So for all those mask wearers that like to leave their nose hanging out, you're leaving your major portal of entry hanging out for the virus to get into. Portal of exit is going to be the same places. So portal of exit is going to be saliva or nasal droplets. So saliva and nasal droplets are the portal of exit. There's some question about whether fecal matter, which can contain live virus, might also be a portal of exit. But in the United States, it's probably not a significant one, meaning not a significant way to transmit the virus from one person to another. Let's look at HIV and AIDS. So HIV is a virus that causes the disease of AIDS. There are actually a couple different portals of entry. There's two major ones. One portal of entry is going to be the mucous membranes of the genitalia. So that could be the urethra of the penis. It could actually be, if you're uncircumcised, there's some cells that are around the head of the penis that could be the portal of entry. The anus is a portal of entry because that's a mucous membrane. And then the vaginal walls or cervix would also be a portal of entry. So another portal of entry is directly into the blood. And an example of this would be a needle. So if you're injecting illicit drugs like heroin, then that is a portal of entry using a dirty needle. Also, blood transfusions. When the AIDS epidemic first started, blood transfusions were a major method of transmission. And so, again, that's an IV, bringing that blood into you. So that's a needle. So portal of entry is a needle prick into the blood. And then finally, newborns can get HIV from their HIV-positive mothers through breast milk. So in that case, the portal of entry is the mouth. The portal of exit is going to be either blood, breast milk, or secretions from the genitalia, semen being the most common one. So lots of portals of exit, lots of portals of entry for that one. It's not as easy as maybe some of the others because it's got more diversity. Okay, let's take a look at Ebola. Let's start with portal of entry. This particular virus basically likes any mucous membrane or blood. So portal of entry can be any mucous membrane that it can find. So that would be your eyes, your nose, your mouth, your genitalia. Those are the major ones. It can also be direct into blood. So there were a couple of people that got Ebola that were scientists working with blood draws and accidentally pricked themselves with a needle and gave themselves Ebola. So portal of exit. Portal of exit is going to be blood and any body secretion that's not urine or feces typically. So semen, breast milk, vaginal secretions, a little bit in the saliva, definitely in the breast milk, definitely in the blood. Not very much in the sweat, although it has been detected in the sweat before. Okay, let's take a look at malaria. Malaria, this one's interesting because portal of exit is going to be specifically done by a mosquito. So mosquito draws blood, and that's your portal of exit. So you actually need a mosquito for this. Portal of entry is going to be into a bite by a mosquito in the skin. So portal of entry is the skin through a mosquito bite. And finally, I put this one up here, athlete's foot. So athlete's foot is caused by a fungus, and the fungus makes little spores that basically accumulate in that cracked area in the skin, and then the spores spread everywhere. So the portal of exit is actually skin. And portal of entry is also skin. So those spores land on somebody else's toes in a shower, let's say of a locker room, and they find their way in and they kind of dig in and create new colonies of fungus that create athlete's foot. So portal of entry is skin and portal of exit is skin. Okay, now that we've taken a look at these, I'd like you to go ahead and pause this video and try to fill these in. I didn't talk about the reservoir, but you can look them up. So again, look up the reservoir for each of these diseases. Try to figure out if they're human, another animal, or some sort of non-living supply. And then go back and rewatch the video if needed and list the portals of exit and entry. Now, I'm not expecting you to memorize these, but I will be giving you examples of diseases on the exam and then asking you from that information to be able to determine the reservoir, the portal of exit, and the portal of entry. So once again, I'm not asking you to memorize any of these. I'm asking you to do this so you get familiar with some of the general portals of exit and portals of entry. Our last topic that deals with transmission is how does that particular pathogen get from one person to another? So the most common way is what is called direct. And a direct transmission is basically that you have a person and you have another person, and then you've got some sort of pathogen. Let's put pathogen, and somehow it goes directly from one person into another. Examples of diseases that are transmitted directly or often transmitted directly would be like sexually transmitted diseases. COVID-19 could be directly transmitted from the aerosolized droplets of one person going directly to another. Measles could be transmitted that way. I mean, lots of stuff is transmitted that way, just from one person directly to another, through close contact, through direct contact, through sexual contact. So something being in this, breathing the same air, that would all be examples of direct. Touching. Indirect usually means there's an intermediate. So an indirect means that you've got a person and they've got a disease. So we'll use the same little red dots here. And then somehow those red dots get to something else, an intermediate of some sort, and then another person somehow picks up those red dots. Now, what is that intermediate? That intermediate can be an inanimate or a living object. For example, a dirty needle is an inanimate object that can serve as a vector for transmitting a particular disease like HIV, Ebola. Inanimate objects that serve as vectors for particular pathogens are known as fomites. So they actually get a formal name. Needles, scalpels, a contaminated catheter, for instance, are all examples of fomites. These are just basically non-living things that are transmitting a pathogen. Sometimes the pathogens require animals as an indirect way to transmit. A lot of these are biting insects. These are referred to as vectors. So these are usually biting insects that transmit the disease from one person to another. Malaria, of course, is a classic example. African sleeping sickness is another. There's a number of different types of pathogenic worms that are transferred by biting insects. So when you see the involvement of animals in the spread of infectious disease, make sure that you can distinguish between an animal vector, which is usually a biting insect of some sort, that is playing a role in the transmission, but they're not actually playing host to the actual pathogen. So vectors are not the same thing as reservoirs. So let me give you an example. Plague. Plague is caused by Yersinia pestis, which is a bacterial species. The reservoir for Yersinia pestis is actually rodents like rats and mice. The vector is fleas. So we have two animals involved. One is the reservoir. That's where the pathogen basically makes its home, usually, their preferred host, as you will. And then the agent that helps transmit the disease from animal to animal. And again, those are usually biting insects of some sort, like fleas or ticks or mosquitoes. That concludes our lecture, finally, on the process of disease. Thanks.